Enamel proteins reveal biological sex and genetic variability within southern African Paranthropus. Researchers have managed the incredible feat of predicting genetic relationships between some of the earliest hominins to live on planet Earth, using little more than some proteins scraped from 2-million-year-old fossilized teeth. The team behind the new study, mostly from the University of Copenhagen in Denmark and the University of Cape Town in South Africa, says the analysis will be vitally useful in tracing the distant family tree of human beings. The evolutionary relationships among extinct African hominin taxa are highly debated and largely unresolved, due in part to a lack of molecular data, write the researchers. Even within taxa, it is not always clear, based on morphology alone, whether ranges of variation are due to sexual dimorphism versus potentially undescribed taxonomic diversity. The research will help paleoanthropologists chart out which differences in the fossil record are down to natural variations between men and women, sexual dimorphism, and which signify separate species of hominids altogether. Flash floods in an otherwise arid area were responsible for these teeth being so well buried and preserved, the team reports. That may limit how many other fossils we can find like this, but the techniques used here should be able to be applied elsewhere too. This study demonstrates the feasibility of recovering informative early Pleistocene hominin enamel proteins from Africa, write the researchers. We anticipate that this approach can be widely applied to geologically comparable sites within South Africa, and possibly more broadly across the continent.